Hello, everybody. It's Mrs. Pound, and I'm back with another chapter, Chapter 6.2, in the application of genetics from purposeful design life science. Today, we're going to talk about applied genetics from Section 621. So, we've talked about genetics, but how do we use it? Our objectives are going to be to distinguish between selective breeding and inbreeding and paraphrase the process of genetic engineering. Humans have been applying genetics for a long time through the process of selective breeding. This is the intentional crossing of plants and animals. In fact, we even see evidence of selective breeding in the Bible when it talks about Jacob and how he was um, getting his income from his father in law by breeding the sheep together to get the best sheep for himself. That would have been an example of selective breeding in the Bible. Parent organisms are chosen for their traits to get the desired traits in the offspring. So, for this little puppy, this is actually a little Chihuahua. Traits were chosen to give maybe certain colors and the size of this little puppy. There's also cross fertilization. This is a type of selective breeding. This produces exceptionally strong and vigorous plants. This is also known as hybrid vigor. Actually, in cross fertilization, you choose plants with opposite characteristics so that you get this hybrid vigor. It just produces better plants when you have the opposite genes, and you know that you're going to get that one very good dominant gene that shows. There's also inbreeding for selective breeding. This is the mating of closely related individuals. This can cause weakened varieties. That's because it not only intensifies the traits that we want, but it also brings out if there are any genetic disorders or diseases. It also brings out the bad traits. So, for example, in certain dog breeds, they have hip problems or blindness and other uh, abnormalities that we don't desire that have been bred in to that particular breed. So that is the danger of inbreeding. Now, something more modern is genetic engineering. This is the process used by scientists to transfer genes or parts of DNA from one organism to another. And actually, even though I say this is new to us doing this, God actually created this because we are just borrowing from some of his design because we use bacteria and some viruses to help us with genetic engineering. And the reason we do that is because they do this naturally. We're just using them and figured out what God already set up. And this can be used to, in a sense, breed organisms from different species. When there are characteristics that we want from organisms from different species, we can then put those characteristics in. One such example is we actually use bacteria to produce insulin for us for diabetics. So, how it works, how do we do things like make insulin? The first thing is scientists decide what gene they want to copy. For instance, the gene that produces insulin. Then a carrier is selected. This carrier is called a vector, and this vector is what's called a plasmid. And a plasmid is a ring of DNA in a bacterial cell, like E. coli. So a vector is a carrier used to get recombinant DNA into the nucleus of a target organism. Now, when I say recombinant, that sounds like recombined. So when I talk about recombinant DNA, it's just DNA that we have recombined. This process that we're talking about here is transformation. This is changing the DNA of cells by using recombinant DNA. Right here is a DNA fragment and a plasmid vector. So this plasmid that I talked about, this ring of DNA from bacteria, makes up our recombinant DNA, and this is transformation. This third step is the gene and the vector are cut into pieces. So they cut the gene out, they cut a slice in the vector, and then they fit them together into the vector. The piece of DNA is fitted into the vector, so we have the recombinant DNA. Step 5 is the recombinant DNA is inserted into a bacterium like E. coli. The reason we like to use E. coli for manufacturing something like insulin is because the E. coli will reproduce about every 20 minutes. So we can produce a very large number of bacteria that are producing a lot of insulin for us that we then purify. So, and then 6, the bacteria makes the protein using the code inserted into the recombinant DNA. Another vector that we can use is called a bacteriophage, and these are viruses that attack bacteria. The reason we use them is they have a simple structure which makes them good vectors, and when we use this, we call it transfection instead of transformation, transfection, because it sounds like it's being infected, which in effect it is. It's infecting the bacteria to make, with the DNA to make things that we want, okay? So, and there are all types of different kinds of genetic engineering that are going on. When we talk about genetically modified organisms like plants and animals, there are different ways that that is done. We insert genes into fruits and vegetables to give them desired characteristics we want, perhaps color or something else. Something that helps it to resist disease, all those kinds of things. We're also looking into using things like viruses, like the bacteriophages, to help us perhaps fight diseases like cystic fibrosis. If we could infect a human who has cystic fibrosis with a virus that would take the bad DNA out and insert it with good DNA so that they're producing the correct enzymes, that could potentially be beneficial. However, we do have to think of the ethics of all of these things. For instance, what if it gets released out into the environment? Are there bad effects for some of these things? So there's a lot of ethics involved when we're talking about genetic engineering as well. So our objectives were to distinguish between selective breeding and inbreeding and paraphrase the process of genetic engineering. So don't forget your 5 questions on your notes, and remember to keep watching this video to answer the questions at the end.